Good morning, Dr. Ravichandran, sir.
Good morning, sir.
Good morning. So, shall we start with the session?
Yeah. No problem.
Yeah, thank you, sir.
No, thank you, sir.
Yes. Yes, sir.
Hello. Good morning. Am I audible?
Yes, sir.
Okay. Good morning, one and all. I, Ms. Ankita Shinde, Assistant Professor, Department of Pharmaceutics, DPU Pharmacy, Pimpri, Pune, feel privileged to welcome you all on behalf of DPU Pharmacy for the fourth session of Third International Faculty Development Program on Emerging Trends in Phytopharmaceutical Research. In the last week, we had our third scientific session on 1st April. It was delivered by Dr. Deepak Kasote, Senior Research Specialist, Missouri University Metabolomics Center, University of Missouri, Columbia, USA. He addressed the participants regarding the use of metabolomics as a tool to check the quality and standardize the drugs obtained from herbal origin. Today, we have with us Dr. Veerasamy Ravichandran, Head Pharmaceutical Unit, Faculty of Pharmacy, AIMST University, Malaysia. He would be delivering a session on the topic modern approaches in natural product drug discovery and drug development. Now, I request Dr. Ravindra Vavare to please introduce our today's speaker. Please, sir.
Thank you. It's my privilege to introduce you today's.
Sir, please unmute yourself.
Yes. Thank you, madam. It's my privilege to introduce you today's speaker, Dr. V. Ravichandran, Professor and Head, Pharmaceutical Chemistry Unit, Faculty of Pharmacy, AIMST University, Malaysia. Sir completed their PhD in Pharmaceutical Sciences and M.Pharmacy in Pharmaceutical Chemistry from Dr. H.S. Gaur, Vishwavidyalaya, India. Sir has many years of experience in the academics and in research. Sir is currently Unit Head and Professor of Pharmaceutical Chemistry, Faculty of Pharmacy, AIMST, Malaysia, and also the Professor, Faculty of Pharmacy in the same university. And as a Sir has many awards and recognitions on their credit. Few of them are appointed as a Deputy Director, Center for Postgraduate Studies, AIMST University, Malaysia, elected as a Senate member, AIMST University, Malaysia, placed in the top 1,000 scientist position in Malaysia in 2022, according to AD Scientific Index for the year 2022, have been appointed as an expert reviewer for NAWA, Polish National Agency for Academic Exchange under the ULAM program. Sir also featured among top 2 scientists in the world in 2021 by Stanford University, USA. Sir placed in the top 1,000 scientist position in Malaysia in 2022-2021 according to AD Scientific Index for the year 2021 and 2020. Sir is also serving as a Director, Center of Excellence in Biomedical Sciences in AIMST University, received PMSDS Distinguished Pharmaceutical Sciences Award in 2021. Sir also received Dr. APJ Abdul Kalam Award for Teaching Excellence in 2020 and Scientific Excellence in 2020. And there are many awards in their list. Sir's major interest in design, synthesis, and evaluation of various medicinal compounds, QSAR and molecular modeling studies to explore structure and activity relationships, phytochemical and biological investigation of medicinal plants, green synthesis and characterization of metal nanoparticles, development and validation of analytical methods and pharmaceuticals. Sir received various research grants. Some of them are FRGS grant from Ministry of Higher Education, MOHE, Government of Malaysia, FRGS grant for Ministry of Higher Education, MOHE, Government of Malaysia in 2018, and AIMST University Research Grant in 2013. Focused on their research outcomes, There are total more than 150 publications which are international and national on their credit. Cumulative impact factor of last 5 years is more than 250. Scopus citation is more than 3,000. Scopus H index is around 26. Google Scholar citation is more than 5,000. Google Scholar H index is more than 30. Google Scholar I10 index is more than 70. So it is our privilege to have with us a great speaker in the area of medicinal chemistry, in the area of phytopharmaceutical chemistry. So thank you very much, sir, for accepting our request and for delivering this session. So I request you, sir, to take forward this session.
Thank you, sir.
Yeah, thank you.
Please allow me to share the screen.
Yes.
My screen is visible, sir?
Yes, it's visible, sir.
Yeah, thank you, sir. Okay, good morning to all. First of all, I would like to thank the management of Dr. D.Y. Patel Institute of Pharmaceutical Sciences and Research for organizing this international faculty development program for the last 3 years. And I would like to thank the principal, vice principal, and the coordinator, Dr. Ravindra, and the chief coordinator, Dr. Asa Thomas, for inviting me to deliver a talk on modern approaches in natural product drug discovery and development. And I'm also thankful to the audience, means the participants today. So with that, I can move to the topic. Just now, the coordinators informed that earlier, the last week, you had a talk on the metabolomics. That is the main concept, what I'm also going to discuss today. But it is a little bit different from the metabolomics also. So today's topic is modern approaches in natural product discovery and development. So first, I would like to tell my disclaimer that the slide contains condensed pictures, data taken from web article, lectures, tutorial, and its respective authors want their copyrights because it's nothing else belongs to me. I have collected all the information from the internet source and the article and books. So the owner of the articles are the copyright owners. Then all other opinions, suggestion, and comments, what I'm going to do during my talk is my personal capacity only. There is no relation with my institution or my superiors here or any others. Everything is my personal opinion and suggestions. And sometimes I'm going to talk about some software or something. It is, everything is my personal opinions. There is no relation between me and the software company or any other agent. So everything is my personal opinion. So I'm going to talk about in 3 main objectives. One is to discuss the importance of natural product research and discuss the challenges and hurdles in the exploration of natural product. The last one is to discuss the role of modern approaches. What are the modern approaches to face the challenges? What we are going to see as our second objectives, how we are going to face and how you are going to solve the hurdles in natural product research. Anyway, I think the last week, FTP itself, the doctor who has given the talk has discussed about all these things. Anyway, so the total research, as we all are aware that because all our faculty members, so you all are aware that there is a number of years and the cost, what we are going to spend to develop a single drug. That is near about 10 to 15 years you have to spend to develop a single drug, whether it is a synthetic drug or a natural product drug. So the minimum years, 10 to 15 years, in that we are going to spend about 5 to 6 years to identify the disease and identify the compound. The remaining years to develop the formulation and You have to go for phase 1 to phase 4 clinical trials and get the approval from FDA. So all these things, the remaining 7 to 8 years, but mainly our concentration is on the middle of the drug discovery. That is, once the disease is identified, you have to find out the lead or hit molecule from either from the natural source or from the synthetic source. That is the main part what we are going to see here, this part, the before the preclinical testing and after the identification of the disease. Here only we are going to spend near about 3 to 4 years. So I think you all are aware that what is the today turnover of the annual worth of the medical marketing. It is near about 1.1 trillion US dollars. So 1.1 trillion altogether, synthetic product and natural product. In that, the natural product itself, it is 35% market it's having. In our medical field, near about 35% is natural product and its derivative. Not only we cannot tell it is only from natural product, its derivative also. So totally 35% of the medical market is covered by the natural product. In that, near about 25% of the drugs are obtained from the plant. Then near about 13% from microorganism and 3% of the drugs from the animals, and remaining percentages are from the synthetic natural product. Means either it is a derivative or it is a synthetic drugs from the pharmacopoeia of natural products. We are going to see all those things later. So these are near about 35%. So 1/3 of the market is occupied by natural products. So you know the importance. So when we are talking about the synthetic drugs, so as I already said, the time and cost is the main factor. When you are talking about synthetic drug, mainly the selection of hit to the preclinical studies and drug discovery. These are the area where we are going to spend a lot of time and money, but it is not much. But when you are coming to natural product, the time and money spending is more. We have to spend a lot of time and the money and the equipment, what we needed. So only nowadays, most of the pharmaceutical industry not showing interest on natural product, even though it is safest, one of the safest medication what we are getting from the natural source. Even though the pharmaceutical company are not showing most important, not giving most important to the natural product. This is the reason, because they have to spend a lot of time. They have to appoint many scientists in many area, not only natural product, medicinal chemist, biochemist, pharmacologist. Compared to synthetic drug, they have to spend more for the natural product research, and the time, money, and all other things. Just you see the graph here. So once the crude extract is taken, after that you see the time and money spent for to develop your natural product is compared to the synthetic product, it is more. So this is the main reason, one of the main reason why the pharmaceutical companies are not giving that much importance for natural product. So as you all are aware that what is the natural product, how we are going to find out that isolate the drug from the natural product, means particularly we are talking about the plants. So first you have to derive the name from the catalog or frequently used plant, means the traditional use, whatever the plants are used in the particular area for a long time, that is traditional use. From that, you have to collect the plant and go for the literature review to collect the information about that particular plant. That is the most important area. First, you have to study what are the works done on that particular plant, what are the compounds isolated from the particular plants. Then evaluate data in literature. That is mainly we are going to concentrate on my talk. This is the database. Then decide on need to test, which test you want to do for the safety wise. Then you have to select the test and you have to perform the test. Before perform the test, you have to prepare the protocol for your bioassay, which bioassay you want to do for the safety and toxicity. Once you have selected the plant and the toxicity proposal, means the bioassay protocol, then you have to carry out the test for safety and toxicity. Suppose from the literature itself, you come to know there is already the published studies for its toxicity and safety, then no need to perform all these tests. So directly you can move to the collection of plant. Once the collection of plants was done, then you have to do the extraction, isolation, and you have to go for the biopsy to find out its biological activity. Then you have to, suppose if that is the first method, by using the extract itself. Suppose you want to isolate the specific compound and do the formulation, then go for isolation and identify active principle, because when you're talking about plants, it's having multiple number of phytoconstituents. Each and every constituent is having its own biological affinity towards the target. So we have to think which one you want. Because sometimes the active constituents A is more potential for what you record. So I want only A. So you have to isolate that compound for your activity. Here only we are going to face a lot of problem how we are going to isolate from the extract. Then carry out human studies and develop method for industrial production purpose. Because the main thing is when you're talking about the natural product, either it is from plant, animal source, or marine source, or any other snake venom, or whatever the source, the yield is very less. So when we need more quantity, it's not possible to collect from the natural source. So that time you have to think about the synthetic route. So that is you have to go for the industrial production process. So the all new approved drugs from 1981 to 2019, the total number of drugs approved by FDA was 1,881. In that, can see and is getting natural product and is botanical and natural derivatives as a synthetic drugs. They are synthetic drugs that is mainly from the NP pharmacopoeia, that is natural product pharmacopoeia, vaccine, and mimic of natural product. So you can see the contribution of natural product here, natural product, botanical natural product derivative, then synthetic drug that is mainly from the pharmacopoeia, then mimic of natural products. So these four area. So you can see the percentage. For example, here you can see the synthetic drugs from natural pharmacopoeia is there are about 65 drugs, 3.2%. And you can see here the natural product derivative is 356 drugs. Out of this 1,881, near about 18.9%. Then natural product drugs are 71. It is contributing near about 3.8% of this total 1,881. Then biological source near about 346 drugs. So here the drugs which are obtained from biological sources called as biological macromolecules, and the drugs directly obtained from the natural source that is called as unaltered. Means we are not going to alter, we are going to use the natural product as like that. That is named as, coded as N. Then botanical drugs, that is the extract mixtures, that is NB. And natural product derivative means here whatever we are getting the products after we are doing the lead optimization, we are going to modify the drugs, means we are going to prepare the derivative of natural product that is coded as ND. And this one is the important thing. Here we are not going to use the natural product directly, or we are not going to prepare the natural product derivative. But what we are going to do here is we are going to take the pharmacopoeia, which is responsible for biological activity. As you all are aware that pharmacopoeia is nothing but the functional group which is responsible for the intended biological activity with its bond length and bond angle. So that is the pharmacopoeia. So we are going to take the pharmacopoeia from the natural product. Suppose it is a very big molecule which is obtained from the natural source. You have to find out which pharmacopoeia is essential for the pharmacological activity. Then you have to synthesize that pharmacopoeia alone, like fragmentation. So you have to select that pharmacopoeia. Best example is the captopril. It is obtained from the venom teraptide. That is the venom, the chemical peptide obtained from the viper venom. So from that later they found out only the one pharmacopoeia molecule is essential for the biological means of the cancer, that is the hypersensitivity that is captopril. So that is one of the best example for the synthetic drug. And mimic of natural product. Sometimes you have to design a molecule which is having the mimicking, means we can go for the stereoisomers or the other drugs which is naturally mimicking the natural product. So we have to design and synthesize the mimic drugs. So these are the things out of this number of drugs FDA approved by FDA. So near about 1/3 is natural product and its derivative. We are not talking about only natural product. We are talking about the natural product and its derivative is 1/3. Near about 35% is contributed to this 1,881 drugs. So this is the drugs approved by the FDA, and in that the biological drugs are 346. And the other small molecules approved by ERS and equal to 1,394. So this radar graph is indicating the year-wise how much drugs are invented by every year, approved by every year. So this is only for the natural product, not only natural product and its derivatives. Just now I told natural product, botanical natural product derivative, and synthetic drugs from the pharmacophore from the natural product. So all together from 1981 to 2018, this is the contribution of the drugs. The different color is indicating different things. The green, dark green is natural product, light green is botanical natural product, the lighter green is natural product derivative, and the orange color is the synthetic drugs. All it's from the natural product. So this is the contribution of natural product, which I have taken from the Journal of Natural Product 2020. And here only the natural product, we are not including the synthetic drugs, only natural product, botanical natural product, and natural product derivative. This is the contribution from 1981 to 2019. And for the last 25 years, if you are taking the FDA approved drugs, both including new chemical entities and biology, this is the contribution. In the year of 2018, there are many drugs approved by the FDA, near about 59 drugs in the last 25 years. And then you can see the year 20 and 21, near about 53 drugs in 2020 and near about 50 drugs in 2021. Why many of the drugs approved in this time? Because of you all aware that due to this pandemic COVID-19, there is a lot of drugs approved for that and vaccine. So only the number of drugs are more in this, particularly this 2 years, 2020 and 2021. So natural products have contributed greatly to drug discovery. Just now we have seen the contribution of natural product in a different way, not directly and indirectly. So it is contributing 1/3 of the medical market. So that is the important things why we are giving more concern to the natural product. The FDA approved new molecular entities revealed that natural product and their derivatives present over 1/3 of all new molecular entities. The world market is, the phytomedicine is covering 60 to 100 billion US. And India is blessed with mega diversity of herbals. So you know, from south to north India, everywhere that is, we are having the big diversity of herbals, and which coupled with research knowledge of traditional use, we are having the lot of what you can tell the literature and the information, that traditional uses of the plants. Means here we are talking about herbal remedies. We are having a lot of traditional method of use of the medicines in different area. So we are having both the information what we need to do the better for the natural product drug discovery. The first one is the herbals. We are getting enough in India, and the traditional use, that is the added advantage. Instead of starting from beginning, if you are having the traditional history, then it is easy. We can go for the development of drug for the particular disease easily from the particular plant what the people are using, traditionally using for the particular issues, health issues. So natural products, it's not only coming from plant and animals, it's also coming from the microbes as well as venom and toxins and marine world. The main source is plant, followed by microbes, animals, marine, and venom. So why we have to go for natural product? Because there is a lot of structural diversity. Structural diversity means, as I earlier I said that the plant, it is contains, there is a lot of constituents.
Excuse me, sir. Hello, Ravichandran, sir.
He got disconnected. Just message me.
Hello, can you hear me?
Yes, yes, sir.
Yeah. Sorry for the interruption, sir. Some internet issues.
No problem.
Okay, the safety and efficacy is most important thing here when we are talking about the natural product. Then the physiochemical properties of natural products comparable with the synthetic drugs, the more carbon, hydrogen and oxygen and less nitrogen. When you are talking about the natural product, it is having more carbon and hydrogen as well as oxygen, but when we are talking about the nitrogen, it is less number of nitrogen, because many of the drugs, when it is having nitrogen, it leads to some unwanted effect. But when you're taking natural product, very few product only it's having the number of hydrogen more. Otherwise, it is having very less number, and many of the product not having the nitrogen. That is the added advantage when we are talking about the natural product. Then evolutionary selected for biological target means when you are taking the natural product, it is having affinity towards many of the target. That is the added advantage when we are talking about the phytoconstituents of the plant and other sources. Then it follows Lipinski rule of drug for formulation, particularly when we are taking the oral drug. When you are talking about the oral drug, it has to follow the Lipinski rule of 5. That is, in as you all aware that the molecular weight P should be less than or equal to 500. And the log P value should be less than or equal to 5. Then when you're talking about acceptor, it should be less than or equal to 10, and donor, it should be less than or equal to 5, and rotatable bond should be less than or equal to 10. So it is a lot of those natural products are obeying this. Then greater water solubility, because we need the solubility both in water as well as the organic solvent. Then only the log P value, when we want the drug should act on CNS, then it has to cross the blood-brain barrier. So that time the hydrophobicity should be more than hydrophilicity. But the natural drugs, when we are going for the oral route, the hydrophilicity is hydrophilicity is most important. So the water solubility is playing a major role in when we are taking a drug in the oral route. Then higher chemical diversity, already we have seen that is a higher chemical diversity, means the number of chemicals available from the natural sources more. So natural product derived drugs means directly we are deriving the drug from natural product. Some natural products derived drugs that are hallmark for modern pharmaceutical care include the well-known drug, anti-malarial drug, that is quinine and theophylline, then penicillin C, the well-known antibiotics, the well-known analgesic is morphine, and well-known anti-cancer drug like paclitaxel, vincristine, and doxorubicin, and antihypertensive drug dioxin, and another antibiotic cyclosporin, and the vitamin A, among many other examples. These are the main drugs directly, which is directly derived from the plant. We are not, they have not done any modification, direct source from the natural product. And curacin A from marine cyanobacterium source, potent anti-tumor activity. It is from the marine source. So the other drugs I have given the name. The other drugs are eleutheropine, then discoderomel, bryostatin, dolostatin, and cephalostatin. These are the other drugs which is obtained from the marine source. Then the other drugs just now I told, which is coming under the synthetic derivatives from the natural pharmacophore, that is captopril. So it is obtained from the teprotide, peptide isolated from the venom of Gracilian viper, lead for the antihypertensive agent, that is captopril. It is the pharmacophore of teprotide. Then potent analgesic compound, that is epipatidin, that is obtained from the skin extract of Ecuadorian poison frog. That is the epipatidin, which is a good analgesic compound. For example, you can see this one, the teprotide, the structure you can see very big molecule. Definitely it's not going to obey the Lipinski rule, all these things, molecular weight, log P value, and at inward donor, etc. You see how many NH2 groups are there and how many OH groups are there, double bond O's there. So definitely it's not going to obey the rule. So instead of using the whole molecule, they have taken this part, only this part, that is captopril. So later they have synthesized this captopril. This is called as the synthetic derivative, means we are not taking the natural product directly, or we are not doing any derivative preparation from this natural product. Instead of that, we are taking only the pharmacophore, which is responsible for the biological action or pharmacological action. So this molecule, they found out this molecule is the responsible pharmacophore for the antihypertensive effect. So they have developed this drug by using this part, that is called as captopril, antihypertensive drugs. Then the marine natural products also and its derivatives are approved. Many drugs are approved for different biological activity from marine source, like for example, anti-cancer, antiviral, cardiovascular drugs, and analgesic drugs. Then challenges in, just now we have seen what are the natural product which is available and which is obtained from different natural sources, whether it is directly from the natural source and the derivatives and its synthetic derivatives. Then what are the challenges in natural products research? You are all aware that the challenges in ensuring the access and adequate supply, because we are going to extract the drug from plant, animal, or insect or mineral source. So how we are going to access? That is the main important thing. Suppose the plant is available in Himalaya hills, but you are in the South India, so you have to go there and collect the drugs for your industry. So the access, it is nearby, then the cost is very less. So the access is most important thing, and how we are going to collect the, or how was the plant material in the morning time or evening. So there is a lot of factors are there, that is ensuring the access and adequate supply. Suppose if you are getting from some sources like from the agricultural or from the market, so the adequate supply should be there for the natural product research. Then complexity of natural product, as already we have discussed about that, the complexity, because whenever you are taking the natural product, it's containing lot of compounds. So we have to isolate which compound is more potential for your desired activity. So you want to separate. So we have to use lot of sophisticated instrument and the other techniques to isolate the compound. So the complexity is the other problem. Then intellectual property right concern, the IP right concern, that is another important thing. Suppose the plant is got patented by someone else, then you are doing work on that plant. So if you find out something, then getting the patent is somewhat difficult. Or the same drug isolated by from some other plant and got it patented. So a lot of things are there. I'm not familiar with this IP rights. So there is a lot of issues when you're doing the natural product research. Then further, the natural products are typically isolated in small quantities. That is the main drawback when we are doing natural product research. Whenever you are doing the extraction, we will get very minute quantity that are insufficient for lead optimization and lead development. So what both the purpose, lead optimization and lead development, we need enough quantity of drugs. If you are not having enough quantity, then we cannot proceed further. That is the main drawback, what the challenges we are going to face in natural product research and clinical trials. So collaborating with synthetic and medicinal chemistry is necessary to determine if the synthesis or semi-synthesis might be possible. Because just now I told the semi-synthesis means we are taking the natural product and do the modification, means you have to prepare the derivatives, that is semi-synthetic, and synthesis when it is the particular pharmacophore of the molecule is the natural product is used as a drug, then you have to think about the synthesis. As a natural product chemist, we cannot do that. So you have to collaborate with synthetic and medicinal chemist. So it is extra manpower and extra money, extra time, everything is involved. So that is another challenge what you have to face in natural product research. The another technique to improve natural product compound development may involve the creation of natural product and natural product like libraries. So when you want to create the libraries, that is the important criteria nowadays. All the scientists they are using, you have to create the library, whether it is synthetic or natural product, you have to create the library. Once the new disease or target is identified, then you have to use the high throughput screening technique. Then you do the bioassay of all this compound against the new disease. So for that, you have to create the library. So creating the natural product library is the another issue. So you have to combine this natural product with combinatorial chemistry. Then only you can develop the big number of compounds in the library. So here combinatorial chemistry, as you all aware that it is the simultaneous synthesis of more than 10 compounds at a time. So by using this, combining this natural product and combinatorial chemistry, then you have to synthesize number of compounds and store it in library for the future HTS, high throughput screening approach. Then this drug discovery from natural sources has traditionally been so time consuming. Just now we have seen in the first two slides itself, how much time consuming is the traditional natural sources, how we are going to extract and isolate the drug from the natural source. Then innovative strategies to improve the process of plant collection are needed, because all the things, many of the drugs already said, near about 25% of the natural products are derived from the plant, either from the leaf, flower, fruit, root, or stem. Means how we are going to collect this material plant parts. That is a major important things. For example, in the earliest days, it's not related with the medicine, just plucking the coconut. Earlier days, the man has to climb in the coconut tree and has to pluck the coconut. But now we are having the climbing motor. So we can use it, and within second you can pluck the coconut. So like that, collection of the tea leaves. Earlier the human has to do. Now we are having the robot to do the collection of the tea leaves. So like that, you have to use some innovative strategies to improve the process of plant collection. That is another important thing. Then screening of extract libraries can be problematic because extract is too complex. So the screening of this library is very difficult. Then challenges in bioassay screening. Even we know the method, bioassay method, but when we are doing the bioassay, we have to face a lot of challenges. That is, all will affect the future drug discovery from the medicinal plant, means from the natural source, not only plant, from all the natural source. These are the some major challenges what we are facing in the natural product drug discovery. For example, you can take your Taxol, that is anti-cancer natural product drug, that was isolated from the park of Taxus brevifolia in the year of 1971, but it took near about 22 years to get it approved. In the year of 1993 only, it has got FDA approval. So near about 22 years. So that is the main drawback in natural product. If you identified also, isolated also, you want to get it approval, there is a lot of procedures you have to do compared to the synthetic drug. It is mainly used in the treatment of all type of cancer, ovarian, prostate, cervical, and pancreatic cancer. So what the main things, what we have to do to solve all this issue? So here I'm going to mainly concentrate on the computational methodology. We are going to use the computational methodology that is instrumental at various stages of drug discovery. In the second slide itself, we have seen what are the different stages of drug discovery. So There we are going to use the computational technology to solve all the issues what we have seen, but it's not possible to solve all the issues. At least we can minimize many of the challenges. We can solve many of the challenges. So computational methodologies are assisting in the exploration of natural product drug discovery. In the stages of discovery, discover new leads more efficiently, then optimize lead, repurpose of known natural product. This is the major developing area nowadays, particularly after the pandemic. So the pandemic, they have repurposed many of the drug for the treatment of this COVID-19. That is, repurpose means the drug which is already approved for some other disease will be utilized for the new target, new disease. That is repurposing. So target new metabolites on the basis of genome analysis. Instead of targeting the metabolites, you have to target the genome. Because already I told, when you're talking about the plant or animal source, it's having number of metabolites, the secondary metabolites, the phytoconstituents. So it's very difficult to do the profiling of metabolites. So instead of that, the metabolites are produced by the genome. So we have to do the genome analysis and find out the particular genome, which genome is going to produce the metabolites. So from that, we can do the drug discovery, that is genomic analysis. Then the computational methodology also useful in revealing the mechanism of action, how it is going to interact with the biological target or enzyme or receptor, how your drug is going to interact. So we can easily understand the mechanism. So these are the different areas where we are going to use the computational methodology in natural product research. So this is already I told, this is the complete the research development process. So where we are going to use which technique. So these are the technique what we are going to use. So our main means when we are identifying the disease, we are going to use genomics, proteomics, and biopharmaceutics to study about the target. Means the new diseases identified, you have to find out what it's happening in your body, whether the enzymes are involved or receptors are involved or proteins are involved or any other things, biological, what are the biochemical changes happen in the body. So all these things studied by genomics, proteomics, and biopharmaceutics. Everything is computer-based technology. Then once the things are identified, you have to find out the drugs. So either you have to go for high throughput screening. For high throughput screening, you have to have the synthetic library. Either it is the library of compounds, either it is a synthetic library or natural product library. Then you have to develop the bioassay, and by using the hyphenated or sophisticated instrument, you have to do the bioassay of the compounds. Okay, that is high throughput screening. Then if you are not having the drug in your means the library containing synthetic or natural product, then you have to go for virtual screening. That is by using software, you have to design the molecule, then do virtual screening by using different techniques like structure-based drug design or ligand-based drug design. By using that, you have to find out the hit molecule, then optimize the hit molecule to lead to drug. That is the another process what we are using. That the same thing we can use it for the natural product also. The main thing here is we are going for dereplication for natural product. That is the area what we are going to see today. That is dereplication for natural product. Dereplication is nothing but the rapid identification of known compound from the natural product extract. So what is the advantage when you are doing dereplication? Dereplication is the term which is used for the rapid identification of active compound, the known active compounds. Why we have to find out the known active compounds from the natural product? What is the advantage? Suppose that known active compounds is highly potential, then we can easily separate that compound and go for the activity, go for the formulation and develop the drug. Otherwise, the other way also you have to think, it is not only to rapid identification of known active compounds. Suppose we want to find out the novel compounds, the other compounds which is showing the pharmacological or biological activity against the particular target. So we don't want to do the same activity for the known compound. So no need to isolate the known compound. So for that time, by using the dereplication technique, we can find out the known compounds. Then the remaining extract, we can use it to find out the novel compounds. So both way we can use the dereplication technique, either to find out the active known compound or the unknown active compounds, that is called as novel compounds. We can find out the new compounds by using dereplication technique. So there we are going to use two methods. One is computer assisted identification of compound. So we are going to use the computational technology to identify the compound. That is done by, one is metabolite guided, the another one is genome guided. So the last week, I think who has taken this metabolomics, they have talked about much about this metabolite guided. And the second one is genome guided. That is what we are going to see today. This is both will be called as the computer assisted identification of compounds. The another one is computer assisted structural elucidation. Means once we have to identify the compound, you have to confirm the compound. So for that, we are going to use computer assisted structural elucidation techniques. So these are the two main techniques which is coming under the dereplication. So apart from that, combinatorial chemistry, once the natural product source is identified, the drug, then we have to prepare the derivatives, synthetic derivatives, or the natural product derivatives. So when you want to prepare the natural product derivatives, you have to go for combinatorial chemistry instead of the conventional synthesis. You have to use combinatorial chemistry to synthesize hundreds of compounds in a day. Then the optimization will done by lead optimization will done by molecular modeling, and all these things. Apart from this, we are using the computational technology to predict the ATMET properties, absorption, distribution, metabolism, excretion, and toxicity. That is also will be predicted by the computational methodology. So in the different stages, it is useful for all this computational technology to minimize or reduce the challenges. So dereplication, the rapid identification of known compounds from natural product. Natural product, the dereplication strategies typically employ a combination of separation science. Means once you've done the extraction, you want to separate it. Just now I told, when you are taking the plant extract, it is a complex molecule. It is containing a lot of phytoconstituents. So you want to separate your compound. So you have to go for separation science. Particularly, you can use any one of the chromatographic technique, particularly GC and LC, gas chromatography and liquid chromatography. That is separation science. Then you have to go for spectroscopic method to do the characterization. You have to confirm the compound. What is a compound? For that, you have to use mass and NMR techniques. Then database, that is the main important things. So you have to separate the compounds and you identify, means characterize the compound. Then you have to compare this compound with database, whether it is known compound or unknown compound. So that is the thing. In dereplication, it is a automated manner. It is in a continuous process. But when you are doing conventionally, first you have to do the extraction, then you have to do the separation, then you have to go for characterization of the compound, then you have to do the manual interpretation. What is this compound, whether it is known or unknown. But here in dereplication technique, it is a continuous automated manner. Everything will be happen in a combination of all these instruments, separation science, spectroscopic method, and database. So at a time, we will come to know whether the compound is known compound or unknown novel compound. So for example, we can take the database scientific and technical network, that is called as STN International, is the largest online repository of natural product structure. So we can get all the information about natural product structure from this network, scientific and technical network. The STN International is a collection of over 180 different databases. So it combined there are about 180 different databases. So that this network, scientific and technical network, you can use this network and go for the comparison. So this is the total natural product discovery process. So the dereplication, just now I told, one is the computer registered identification of compounds. This is metabolite guided. The another one is genomic guided. The next one is computer assisted structural elucidation, that is called as CASC. This is CAIC, CAS, and TICE. So these are the different techniques what you are going to use, mainly in lead discovery and optimization. These are the technique what you are going to use. One is structure-based computer assisted drug design and the ligand-based drug design. Here ligand tracking, pharmacophore modeling, and molecular dynamics. and ligand-based QSAR pharmacomodeling and ligand-based virtual screening. What are the techniques? And at MET, we are going to use the computational technology to predict the pharmacokinetic and toxicological properties. For example, you can use ATMET2, the online tools, or Swiss ADME is there. So you can use any one of the technique. And for here also, there is a lot of softwares are there to do all this technique. So first we can talk about the database, which is the most important techniques here. The database, because once you identify the compound, characterize the compound, then you have to identify whether the compound is known compound or unknown compound. So for that, we need the database. From the database only, we are going to collect the information and compare with your identified compound. So specific databases are natural products are available with physical, chemical, and biological properties used for natural product drug discovery and dereplication process. So for computer registered drug design procedure, database must provide the compound with their molecular structure in chemical file format, bioactivity data, and biomolecular targets. So these three are the important things when we want to do computer registered drug design. You have to get the chemical file format, the biological activity, whatever the activity obtained from the cell-based assays, and the biomolecular target. So dereplication database must cover extensively the chemical and biological space of known natural product. If you know the chemical and biological properties, then only it is easy to compare with the separated and characterized compound. What are the chemical properties of isolated compound? And you have to compare this one with the library compound. So if it is matching, then the isolated compound will be that particular compound. So it is known compound. So other commercially available database for natural product dereplication or the Chapman and Hall's Dictionary of Natural Product, which is used for bioactive natural product database. The marine natural product database is Marlin-Lit. Apart from that, we are having DREP. These are some database which is used for matching the isolated compound with the database. Then those of particular interest in natural product chemist or chemistry or the registry, Napralet, Palestinian, Matline, and Biosis. These are the some other the databases which is used by the natural product chemist. So these are the some databases which is available, essential features of selected database for natural product, particularly based on dereplication and computer assisted drug design. Particularly, you can see here the fourth one that is suitable for dereplication application, for particularly the superscript with the 4 from here to here. All it is used for the dereplication purpose. And if you want to do spectral data comparing, then the compound with the 10. This is 10. These two are for spectral data. So like that, if you want to do the extract, we are not going to isolate the compound. Only we are going to compare the extract. You are having the extract, then you have to compare what are the spectral data of this extract with the database, then go for these two database, particularly then NAPCAR and PCAR, natural product care. There it is mainly giving 2,566 fractional extracts are isolated. That we can easily compare with the data source. So these are the different data source. But here the CAS is not used for dereplication because it's not having any spectral data. It is not related with the spectral data. It will provide only the taxonomy and bioactivity of the compounds. So it is not useful for the dereplication purpose because all this database we can check, which is not giving the spectral data, will not be used for the dereplication technique, because the dereplication just now I told first you have to isolate the compound, means extract, separate the compounds, then you have to go for spectroscopical characterization. That characterization only it is compared with the database. When the database not containing this, then we cannot use these databases for the dereplication purpose. So substructure searching is available for all database. Substructure means the part of the structure. We can easily do with all the database what are given in the table. Then commercial one is CAS, SCI binder, Rexis, ACDNMRDB, and DNP. These are the some commercial softwares. So just now I told the CAS is your largest online database, but not suitable for dereplication because it's not having any spectral data information, NMR or mass spectral data information. Then the other databases mentioned in the table are free. Apart from these databases, all are free. So already I told the natural product care will be used mainly in the when you are having the extract, not the isolated compounds. So PubChem, it is a well-known database which is containing both synthetic and natural product. So we can get a lot of chemical information, and it is one of the largest repositories of natural product. Then Zinc database, this is also well-known database for when you are doing the computer registered drug design. We will get the structural information and chem information, means the chemical information from this database. So these are the well-known databases, PubChem and Zinc database. So this is the process. This is the without replication and with replication. So with replication, what we are going to do, that is putative, that is recognized rapid identification of previously isolated compound in an automated manner. But in without replication, we are going to do the interpretation and the structural elucidation, everything is manually. But here it is a automated manner, means all it will happen continuously. Once the extract will come, it will go for separation. Once it is separated, automatically it will move to the spectroscopical determination, either NMR or mass. Then it will be characterized. Then it will be compared with the database. So that is the advantage of the dereplication technique. That is your automated manner. So dereplication methodologies, as I already said, two technologies is that one is computer adjusted identification of compounds. The another one is computer adjusted structural elucidation. Both are important. One is important for identification and identification of compound. The another one is important for structural elucidation. These two are important in dereplication because we are going to find out the compound is already known compound or a novel compound. So for that, you have to identify the compound, then you have to compare the compound with the database. For that, you have to need the structural information, that is structural elucidation. So first, metabolite guided. So I think you all heard about this technique, metabolomics. Already it was discussed in the last Saturday talk. So the same thing here we are going to see, that is metabolite guided. It is nothing but the metabolomic approach. Means you are going to study about the metabolites profile. How many metabolites are present in your, what is the individual metabolites present in your tissue or organism? That is metabolomics, what you are going to study. Suppose if you are doing some comparative study, then it is called as metabolomics. So different analytical techniques like all the hyphenated techniques. For example, the mass hyphenated techniques are LC liquid chromatography mass spectroscopy, liquid chromatography high resolution mass spectroscopy, liquid chromatography time of flight mass spectroscopy, then HPLC UV ultraviolet mass spectroscopy, then electrospray ionization Fourier transform ion cyclotron resonance mass spectroscopy, size exclusion chromatography, electro spray ionization Fourier transform ion cyclotron resonance mass spectroscopy, high resolution electrospray ion spectroscopy. Then when we are coming to NMR, 1D and 2D, 1D NMR, you know, all know that carbon 13 NMR and proton NMR. In 2D NMR, total correlation spectroscopy, then heteronuclear single quantum coherence spectroscopy, heteronuclear multiple compound based spectroscopy, then liquid chromatography NMR, then liquid chromatography NMR mass, liquid chromatography solid base exclusion nuclear magnetic resonance spectroscopy. So these are the different spectroscopical hyphenated techniques which is used in the metabolite identification. Why we have to find out the metabolite? The metabolite is, what is the nature of the metabolite? Means we want to elucidate the structure. For that, we are going for all this technique, followed by multivariate data analysis. That is the important things to minimize the redundancy in the isolation steps. So either you have to use artificial neural network or principal component analysis to use that, and you have to minimize the redundancy, means the repetition in isolation steps. So this is the common technique, what it is used in the metabolite. So first you have to take the sample and you have to inject into the HPLC column. Once the outlet coming from the HPLC, it will be go to the mass detector as well as the fraction collector. So the mass detector is used for these things. It means you have to elucidate the structure. So it will be connected with the mass detector, and the remaining things will go to fraction collector. Because you have to collect the different fraction, then only it is easy which fraction is that particular compound is present. Then we can go for the biological activity. The same thing here, it is another representation. So for example, the Chanana et al. Developed an LCMS based principal component analysis workflow, which comprised a new script written in R that is used to distinguish unique versus common metabolites in there are about 15 marine actinomycetes strains. So they have used this technique to distinguish unique versus the common metabolites in 50 marine actinomyces. The database what they have used is antibase. Database was used in this workflow. By using this technique, means the separation technique is LCMS, and the comparison technique, the statistical technique is multivariate analysis, is principal component analysis, and the database, what they have used is antibase. With this strategy, the author were able to find out the skeleton of furosolene, the chemical compound furosolene, one of the 3 classes of novel compounds previously identified from an actinomyceta species. It is a well-known compound, already known compound, but when they are doing the replication studies, they found out this species are containing the furosolene. That is rapid identification. For rapid identification, they have used the dereplication technique. That is the one example for how to rapidly identify the known compound from the new natural product extract. So this is the chemical structure of antifungal agent, that is furosolene 1 and 2 isolated from actinomyceta. That is, one is compound 1 is having the R position is Cl here, and the second one is having Br, that is compound B. The same time, the other scientist Abdul Mohsen et al. Used the same technique, principal component analysis and hierarchical clustering and orthogonal partial least square discriminant analysis to evaluate the high resolution Fourier transform mass spectroscopy and NMR data of marine sponge associated with bacterium Actinokinospora crude extract. So which were cultivated from the Red Sea sponge, Spongia vagabunda. With this dereplication workflow, 2 new compounds. The earlier one is the known compound. They fit identification of known compound. Just now we have seen in the previous slide, but here they have identified 2 new antiparasitic orthoglycosylated angucyclines, that is actinosporin E and actinosporin B were identified. So what is the things what I told? The dereplication technique is your automated manner process to identify rapid identification of known compound or the novel compound also. So both are possible. So we can easily rectify the challenges what we are facing when you are doing the extraction and the isolation and characterization of compounds by the conventional method. So challenges in metabolite guided replication, that is metabolomic studies. Commonly it is called as metabolomic studies. So comprehensive mass and NMR database are not available. That is the main drawback. Yes, when you are isolated compound, you have done the spectroscopical technique, we are having the data, but there is no data available in the database. NMR and mass spectral databases are not there, then we cannot compare with the things. So that time it's very difficult to manage the things. Then most databases do not contain mass fragmentation, that fragmentation spectra and 2-dimensional NMR spectra. They are having mainly the mass spectra as well as the 1-dimensional 1D NMR spectra, not 2D NMR spectra. 2D means toxic, OC, NOC, all it is called as 2D NMR. It's not having the 2D NMR spectra. Then it is very difficult to identify the compound. So these are the main challenges what we have to face when we are preferring to go for metabolic guided dereplication. Then in silico identification software, which generate in silico spectra of lipids, means whenever we are not having the spectra of NMR spectra or mass spectra of the particular compound, then you have to use the software to predict the spectra by in silico. of the purpose of structural identification. So we can solve the issue what we are facing here, but it's not 100% perfect when you're doing this with the artificial intelligence. This method uses artificial neural networks to find accurate bond cleavage rates in mass spectrometer, employing collision-induced dissociation tandem mass spectrometry to develop the spectra. Then searching a database of 18,399 calculated spectra against the experimental spectra of 45 test lipids yielded the correct structure at the top position in 40 cases and at the second position in 5 cases. So the spectra developed by this technique is useful here. When you are not having the spectra, then you have to use this in silico identification software to create the spectra of the compounds. Otherwise, you can go for the finger ID. The fingerprints of mass spectra will be used by using the machine learning approach. Then the predicted fingerprint can be used to search for the unknown molecule in chemical structure database later on. Then for example, you can use the support vector machine for the search. So this is the metabolomic workflow. I think already the doctor who has taken this metabolomic studies, you have studied this flow. So the same flow which is used in the replication. Then genome guide. So the problem is in the metabolite guided studies. So as I already said, when you are taking the natural product, it is having number of compounds. The complexity is more, number of metabolites are present. So isolation, characterization is very difficult. So instead of that, what you have to do, you have to study the genome. The genome is the genes which is going to produce the metabolites. So we are going to study the genome. So once you confirm this particular genome is present in that particular tissue or plant or organ, so it is going to produce these are the metabolites. So we are having the genome database. So comparing this genome with genome database, we can easily come to know what are the genomes present in your particular plant or extract. Then we can easily find out what will be the metabolites. So we can easily come to know what are the metabolites that's going to be produced by that particular plant extract. So the fast development of genome sequencing method means changed the natural product research in spite of the large diversity of secondary metabolites. That only I told there is number of thousands of compounds. The structure of the involved enzyme are much conserved, means it is very less the enzyme involved in the production of metabolites. Very few enzymes are there, making it possible to mine genomes for genes encoding biosynthetic enzyme. So we are going to use the genomic sequence of the enzyme for find out the metabolites, what it is going to produce. So over the last decade, a few hundred molecules have been discovered using genome mining, and many of those molecules were so challenging to discover that the process would be difficult to generalize and automate. By the metabolite method, it's very difficult to go for the automation. So those type of molecules are isolated by genome mining. So genome mining study reported the identification of 31 cyanobacteria gene clusters from 126 genomes of the marine cyanobacteria, that is Microcystis aeruginosa and Oscillatoria nigroviridis. So cyanobacteria exhibited cytotoxic activity against cancer, cell lines, as well as antiviral, antimalarial, and allopathic activities. So the bioinformatics studies play a major role. To analyze the genomes, predicted that the strains produced cyanobacteria with chain length of 3, 4, and 5 amino acid and containing thiazole. So these are the condition the enzyme, the particular genome will produce the cyanobacteria with the chain length of 3, 4, and 5 amino acid and containing thiazole. Particularly, it is indicating the cysteine molecule. So that is responsible for the cyanobacteria synthesis. So it is easily identified. The computational identification of genes encoding metabolic pathway is the fact that they are typically chromosomally adjacent and forming biosynthetic gene clusters. So once we will come to know this biosynthetic gene cluster, then the cluster will be compared with the database, gene database. Then we can come to know what are the metabolites going to be produced. So they are useful targets for mining genomes based on knowledge of homologous genes and roots patterns extracted from them. So we can easily compare with the database. So plenty of computational tools are available for researchers to mine the genetic data, same like what you did in metabolic data. For genetic data also, there are a lot of computational tools are there to do and to connect them to known secondary metabolites. Means suppose you are identifying one gene, the gene is going to produce what are the metabolites. So we are having the tools, computational tools to find out the genetic data and which is connected with what are the secondary metabolites produced by the particular gene. The secondary metabolites bioinformatics portal, that is AIMST website, maintain a catalog of available software database and hand-curated links to major resources used in this field, particularly in this field, the genome and comparison with the metabolites. We can use this website as AIMST. And PRISM, the another database, is an open source application for the genomic prediction and dereplication of non-ribosomal peptide and type 1, type 2 polyketide chemical structures. So that is specifically for the peptide comparison, the gene which is producing the non-ribosomal peptide. For that, you can use this PRISM applying tool. So this is the role of computational methodologies in genome mining for natural product discovery. So these are the genomic and metagenomic space. So these are the different area what we are going to use this identifying biosynthetic gene clusters. So summary of particular features of dereplication workflow. There are 5 steps are there. The first one is identification of the major compound in a single extract. Means we want to find out the major compound, whether it is known or unknown compound. Particularly it is a known compound. So this is the more potential compound. We want to identify that major compound in a single extract. So that is called as the DREP1. Then DREP2, acceleration of activity guided fractionation, means based upon the activity, you have to do the fractionation, different fractionation of compounds. It is not single compound, it is multiple compound, but we are going to do the fractionation based upon the biological activity. The third one is chemical profiling of crude extract collection. It is a crude extract collection. We are going to do the chemical profiling, metabolite profiling. The fourth one is chemical profiling of target compounds. Suppose if you know the target compounds, then you are going to do the chemical profiling of that. The last one is the genomics, that is taxonomic identification of microbial strains, that is coming under gene database, and other things are compared with the metabolites, because all are, it is related with the metabolite, whether it is a chemical information, single compound, or the compounds based on biological activity or chemical profiling. Everything is related with metabolite database. So here, what are the things are given? Means like a targeted chemical class, either yes or no, and sample, single extract, extraction, collection, different things, what is happening in the different stages. So dereplication of natural product, one concept involved in different strategies. So here, just now we have seen the DREP1 is identification of the major compound in a single extract. So that is DREP1. So we are going to take the natural extract, going for fractionation by using different chromatographical techniques. We are going to do the, first you have to collect the different fraction. Then you have to do the structural elucidation by untargeted analysis, because it is not based upon the pharmacology. We don't know anything. Just you have to do the simple analysis by using any one of these techniques. Then the metabolite compare, means from after elucidation, we come to know the structures, then compare with the metabolite database. So then you can find out rapid identification of the major known compounds in a single extract. So here the fractionation is only the step. After doing the extraction, you have to go for fractionation. Then the fractionated material, extracted material will be undergo for the spectroscopical determination, either any one of this technique. Then once you will get the information, then it is compared with the metabolite database. So that is called used for the rapid identification of major known compounds, that is DREP1. Then this is based upon the untargeted analysis, means we are not targeting any biological activity, but known compound. But here it is related with the biological activity screening. So first you have to go for fractionation, then you have to do the biological test, means which fraction is responsible for the particular activity. Then you have to do the analysis of only active fraction, because here we are having many fractions. Suppose this third fraction is having the activity against, suppose I'm doing the screening for anti-cancer. So only the third fraction is showing anti-fraction means anti-cancer activity. Then I will select only this, then go for analysis of that. Instead of going analysis of all the fraction, I will go only this one because this fraction only it's showing anti-cancer activity. So you do the analysis by using any one of this technique, then compare with the database. So you will come out with known active compounds. Okay, suppose you don't want to do the biological test, then go for the fractionation and analysis all the fraction. Then you have to compare with the metabolite database, then you have to go for biological tests. So this way also you can go, but this is the best way to find out which one is the active fraction instead of going for this. This is called as targeting novel compounds. Yes, we are finding out which compound, the known compound. Then you have to find out its biological activity. This is not like that. Any fraction which is showing activity, then you can do the analysis, then you can identify the compound. That is the difference between the targeted one and non-targeted one. Next one is the collection of natural product, go for fragmentation. It is direct or untargeted analysis both. Then go for statistical pattern recognition, then compare with the metabolite database. It is a extract, the total extract, collection of natural extract. We are not going for further fractionation like here. So high throughput chemical profiling of natural extract collection. So that is the things what we are going to do. We have to collect the different extract and do the statistical pattern recognition. Just now we have seen like principal component analysis or hierarchical cluster analysis. Then you have to do metabolite comparison. The same thing for natural extract, one or several samples, then go for fractionation or not. Then if you are going for fractionation, then do targeted analysis, particular targeted analysis, then compare with the things that is called as target identification of selected molecules. If you are not fractionating, then go for the another way. This is for natural extract, the common extract without any further fractionation, go for cluster analysis and compare with the database. So these are the different step, means a different process in dereplication, the different methods, not steps, different methods, 1, 2, 3, 4, and 5. So computer assisted structural elucidation, that is identification of compounds. What we have seen up to here, that is identification of compounds. Now structural elucidation, how to do. So fully automated structural elucidation from spectroscopy data for small organic molecules from 1D NMR or 2D NMR data. Particularly when it is single molecule, then go for 1D, either proton or NMR. When it is complex molecule, then you have to go for 2D NMR like COSI, TOXI, ROSI, all these things. So computer registered structural elucidation except spectrum have been developed for over 40 years. Currently available packages include the open source. Already we have seen ACD source, LSD, and CMC source. For example, the scientist Troch Pescuria et al. Reported enhanced computer registered structural elucidation procedure for the determination of the relative configuration of natural product, which starts from the molecular formula and combines conventional 1D and 2D NMR spectra with the residual dipolar coupling and or residual chemical shift anisotropy. That is the technique what they have used. The employment of RDC data in conjunction with CAS program automated the determination of relative configuration in molecule of medium complexity and moderate degree of flexibility such as naltrexone, 10-epi, 8-deoxychemampirin, strychnine, and euparnomorin, yohimbin, and N-methylcotin. These are the compounds isolated by this technique. So this is the structure of the two things what they have identified. And next one is the computer assisted drug design in natural product. Once you find out the natural product is active against the particular disease, it's showing affinity, then you want to do the lead optimization, means you have to improve the potency. So that time you have to use any one of the techniques, either it is a structure based drug design method or ligand based drug design method. By using that, you have to optimize the potential to, that is called as the lead optimization. And if you want to increase the pre-formulation characteristics like increase the solubility or log p value, so everything will be done by the help of the computer or either drug design. Apart from that, you can predict the AD ADMET properties, absorption, distribution, metabolism, excretion, and toxicological prediction. So these are the two techniques which is used in computer-aided drug design. I think you all aware about this receptor-based drug design. When we know the receptor structure, the target structure, then we can go for receptor-based drug design. Either it is docking or pharmacophore modeling. Two techniques are there in receptor-based drug design, including the molecular dynamics also. But when we don't know the structure of the receptor, then you can go for ligand-based drug design, either QSAR. In QSAR itself, two things are there, 2D, 3D QSAR, and you can go for pharmacophore modeling. So these are the technique which is used mainly in the natural product to optimize the lead optimization. It is not going to do anything with the extraction or isolation or characterization, but it is used in the lead optimization. Means when you want to prepare the natural product derivatives, that time you can use it. Or we want to do the synthetic derivatives from the pharmacophore, we can use this technique to optimize the properties as well as the toxicities and the kinetic properties. So all the way we can use this one, the computer assisted drug design technology in different way, receptor based or ligand based. So when we know the structure, then we can go for structure-based drug design. The structure means structure of your target protein or enzyme or receptor. If you don't know, then go for ligand-based drug design. So these are the different techniques which is used in the natural product drug design. So information about target, then go for ligand talking or de novo design, molecular dynamics or pharmacophore modeling. When we are not having the information about the protein structure, go for quantitative structure activity relationship or pharmacophore modeling. So all these things used for optimizing the existing lead. Means once you isolated the lead from the extract, then we can use this technique to optimize. Means optimize the potential, optimize the toxicity, optimize the pharmacokinetic properties. We can use this technique. So it is easy. Instead of doing manually, it will take a lot of time and manpower. So we can use it this one. So the ligand-based drug design, all or it is machine learning algorithm to establish QSAR and predict the properties. So best example is QSAR, quantitative structural activity relationship. When you want to do quantitative structural activity relationship for natural product, you need the compound structure along with its biological activity. So then you have to do the regression analysis. Then you have to compare which biological activity is going to affect your pharmacological activity. So for that, we can use the QSAR. QSAR is mainly used for lead optimization, but then lead identification. This is not the technique which is majorly used for lead identification. This is the best technique to do the lead optimization. Either it is 2D QSAR or 3D QSAR. So there are different techniques are there. So this is 3D QSAR. Instead of here, we are going to correlate the physicochemical properties with the biological activity. The 2D parameters will be calculated from 2D structure. So when we are going to use the 3D structures, means the energy minimized structure, then we are going for three-dimensional quantitative structure activity relationship, like you can take pharmacophore modeling or COMSIA, MRA, MSA, a lot of techniques are there. So here we are going to find out the 3D parameters, which is energy related parameters, like how many hydrogen bond donor should be there, hydrophilic, hydrophobic interaction, all these things you are going to study here. So this is also one of the best technique to do the lead optimization in natural product drug delivery. So pharmacophore, already I told pharmacophore. Pharmacophore is nothing but the functional group which is responsible for the biological activity, along with that the bond length and bond angle. So to find out that, we are going to use the pharmacophore technique. So we can easily find out the molecule where it should have hydrophobic group, hydrophilic group, hydrogen bond donor or acceptor. So once you come to know, then we have to introduce the group to improve the potential, or where we don't want that group, we can remove those groups so we can reduce the toxicity. So for example, Dinesh Kumar et al. Performed the target prediction for sporolide A and B using ligand-based pharmacophore screening against known inhibitors and drugs. So they found out 8 pharmacophore features were identified in sporolide A and B, 6 hydrogen bond acceptor, 1 hydrophobic groups, and 1 aromatic ring. The 3-dimensional models were generated and the pharmacophore pattern was used to screen the public binding database with 400,000 known ligands. So this is the advantage of the computational technology in natural product lead optimization. So some other example also I have given here. So these are some chemical structures they are identified by the molecular pharmacophore modeling. Then challenges in ligand-based drug design often contains macrocycles, which are problematic structure for computer-aided drug design due to their size. It is more than 50 Dalton. So it's very difficult to do with the ligand-based drug design. Then drug interaction with multiple target is a cause of drug side effect, because we cannot predict by using the ligand-based drug design where it is going to interact, because each and every molecule is having different functional group. So it is going to definitely it's going to interact with a different target. So when it is the record target, then there is no issue. If it is going to interact with some undesired target, then it's going to produce the side effect or adverse effect. So we cannot minimize that one by using ligand-based drug design. Then network-based approaches have also been used for systematic identification of drug target interaction and assessment of drug safety profiles. That is network-based, means we can easily find out the multi-targeted, what is the main drawback here, interacted with multiple target. That one we can study here. The Pang et al. Proposed a statistical network model to predict new drug target and anti-cancer indication of natural product. So for example, the global drug target network was reconstructed that linked molecule substructure and targets and resulted in 7,314 interaction connecting 751 targets and 2,388 natural product. So some other examples are given here. Later on, you can go through. So then next one is structure-based drug design. So once you know the structure of the target, then you have to go for natural product drug design. The best example is talking and molecular dynamics. For that, you need the extracrystallographic structure of your target. If it's not available, then you can go for NMR structure or electron microscopic structure. If not available, then you have to design by yourself by using homology modeling. Then you have to do the docking studies by using the software. There is a lot of softwares are there. The well-known softwares are Autodoc, Vina, and Schrodinger's. They're having some softwares to do the docking studies. VLife, it is there. So docking, there are two types of docking is there. One is target fishing. The another one is the ligand fishing. Ligand fishing is the normal docking. Means normal docking, we are having only one protein. Then multiple ligands are going to talked with your single protein. That is called as ligand fishing. That is normal docking. Just it is opposite to this. That is called as target fishing. That is reverse docking. Means only one drug molecule will be there. You have to find out the interaction of this molecule with multiple what you can tell the proteins. So that is called as reverse docking or target fishing. So we can find out one molecule how it is going to interact with the different proteins. Then we can find out the different targets. So single molecule, that is multi-target approach. So we can find out the same drug is utilized for different type of disease. That is possible in reverse docking. Here multiple drug with single protein. So some examples I have given how the used in natural product, particularly so reverse docking, the ability of reverse docking for target fishing of a marine natural product was evaluated by Chen et al. Using 40 marine compounds with known anti-tumor activities and known target proteins, but without their crystal structure. Determined a database of anti-tumor protein was constructed with 470 crystal structures corresponding to 150 different target proteins. That is the advantage of reverse docking. After docking the 40 marine natural products to the proteins in the database, it was observed that the predicted binding energy for a given ligand to its known target is usually not the lowest. 55% of the compounds have their reported target ranked in the top 20 and 30% in the top 10. It is noted that the compounds may have multiple targets and some of them may have not been discovered and reported yet. Because one compound we are having, it is interacting with the different compound, different target. It is not identified. That is easily identified when we are going for reverse docking. So challenges in structure-based drug design, the crystallographic structure of protein. If it is not available, then you have to design yourself. That is the main drawback. Then dynamics. So it is a type of simulation studies, same like molecular docking. but it is associated with the time. If it is particular time, how will be the interaction, the initial and after 10 minutes or 10 nanosecond or 20 nanosecond, what will be the complex stability and the interaction molecules. So all these things will be studied by the molecular dynamics. So all these techniques which is used in virtual screening, when we are having number of compounds or more, we cannot do one by one, either talking or these things. So you have to go for the virtual screening. Virtual screening means simultaneously and continuously you have to screen multiple thousands of compounds in a row by using the software, either structure-based drug design or ligand-based drug design. So it is by name itself, it is called as pharmacopoeia-based virtual screening and ligand-based virtual screening and structure-based virtual screening. So these are the techniques. When you are having number of compounds, see here library is having 10 to the power of n molecules. When you are doing all this filtration means you are going to filter out by using different techniques. First is you are using docking, then KSAR, then pharmacophore, then ADME properties finally entered with only n molecules. So we can reduce the number of molecules. So this is pharmacophore-based virtual screening example. Then high throughput screening, when we are having the natural product library. So now new drug is identified. So we know the things. So you have to develop the bioassay method. Then you have to check all the natural product against the particular disease by using the automated technique, that is high throughput screening. In earlier you have to do one by one, but here we are going to use the sophisticated instrument like what we have seen, the hyphenated instrument. So we can check continuously per day, millions of compounds will be checked by high throughput screening. It is not like conventional method, one or two compounds. Then the drugs are also isolated from venom and toxin based drug delivery. So characterization of venom, already we have seen different techniques, particularly the mass and NMR. Then you have to isolate, and purification will be done by gel electrophoresis chromatography techniques like reverse phase HPLC. Then sequencing and structural determination of isolated peptide by using mass NMR X-ray spectroscopy. Then we have to do in vitro and in vivo studies. Then computational system biology approach. So we can use computational approach for each and every stage of drug discovery, like biological and system approach will be implemented to calculate the accurate binding pathway and target the structural function of peptide with the target membrane receptors. So this is the total natural product drug discovery, medicinal plants and microorganism. So what is the innovative extraction method, innovative method, and omics technology. Omics technology is particularly to study about the target like genomics, proteomics, all these things will come under, also metabolomics. So these are the reference what I have used for my slide presentation. I have collected from all these sources. So thank you, Anantal. Now I hand over the session to the chairman.
Thank you very much, sir.
Thank you,
It was
sir.
a really informative session.
Thank you, sir.
Now I request to the participants, if they have any question, they can post in chat box. So we can take those questions in question answer session. Okay, in meantime, sir, we have some questions from our end.
Yeah, sir.
So it is really a wonderful information about the natural product chemistry.
Thank
Sir,
you, sir.
we'd like to know your opinion on natural product repurposing.
Yeah, repurposing it is recently it is a well-growing area, sir, because repurposing is most important, because earlier if you are having one drug, we are using it for only one purpose, but it's not two, because we all know that the drugs on the one drugs are having different functional group. Definitely it is going to interact with many. For example, in synthetic drug, I know the best example, the thalidomide. It was withdrawn because of a teratogenic effect. But now they found out it is very good reagent for anti-cancer. It is a very good anti-cancer drug, and it is also used for some other purpose. I forgot the exact purpose. Now it is again FDA approved that same drug, but we should not use it for a pregnant woman. So that repurposing is most important, particularly natural products. Yes, we know the safety of natural product.
Right, sir. There are various traditional medicines available and they showed miraculous effects.
Yeah,
But those same medicines are using for the same purpose for a long time.
yeah.
So can we through molecular modeling or through other techniques, can we find some novel targets for those molecules?
Yeah,
And
definitely.
what,
Particularly, you can use the reverse talking processor. So we can
Right.
easily find out the other use of that particular drug. Then we have to go for the optimization because it is mainly used for suppose anti-cancer. We want to use it for anti-HIV. So we want to mask that anti-cancer effect. So you have to do the drug optimization, lead optimization. Then we can come with a good target for a viral anti-HIV or something.
Right, sir.
Yeah.
Sir, another question is in the natural product, when we administer those natural herbal medicines, there are multi-components and they
Yeah.
shows some synergistic effect or combine effect. So
Yeah.
when we do the theoretical study of these same molecules,
Yeah.
so how much it is feasible that generally in the molecular modeling like docking study, we done the interaction study of one-to-one molecule.
Yeah.
But here the multiple molecules and their effect in biological system. So how we can mimic them in theoretically?
Yeah, theoretically, it was somewhat difficult, sir. We cannot do the multiple component with a single target simultaneously, but we can do separately. But the synergetic effect, that only the main drawback for our computational technology. We cannot mimic exactly what is happening in our body. We can mimic 60 to 80%, not 100%. So there are few drawbacks. One of the few drawback is this one. We cannot find out the synergetic effect.
Right. Thank you, sir.
Yeah,
so there is one question in chat box.
Yeah.
Where can we do the high throughput studies?
That is where we are having the facilities to carry out the automated manner. We have some laboratories that are having that HTS facility.
Okay.
But in India, I don't know.
Okay,
but
sir.
Many of the institute they're having this HTS in some facilities. There we can,
Right.
but we have to invest a lot. That is the main drawback when you are going for HTS.
Right. So another question is about side effects of herbal medicine.
Yeah.
So can we reduce by these side effects by any approach?
Yeah, that only I told. First, you should know the mechanism. If you are doing extract a whole, it's very difficult. Just now you told the synergetic effect, and there is a lot of complex molecules are there, so it's very difficult for that, sir. But when you are going for isolated compound, then we can use the computational technology to reduce the side effects. But when you are using as a whole, then it's very difficult. Yes, it's very difficult to identify the mechanism when you are using as the extract, the whole extract.
Right. Just a minute. Okay. Yeah. Thank you very much, sir. Sir, we have with us Dr. Adepalli, Research Director of DPU Pharmacy. So I request sir to have interaction with you.
Yeah. It was. Am I audible?
Yeah, yes, audible, sir. Audible, sir.
Yeah, Dr. Ravichandran, it was simply a lucid explanation. I think it is every chemist can be benefited by your total presentation.
Thank you, sir.
Whether it is natural or synthetic, you made all the insights coming together for a researcher to begin with how you should take care of. And that is a wonderful thing. We are very thankful to you.
Thank you, sir.
Number
Thank
one,
you, sir.
Yeah, I think when our Ravi asked you about the repurposing,
yes,
you
sir.
said thalidomide. Yes, it's very good. And it is repurposed now as an immunomodulator.
Yeah, sorry, I forgot that one.
Okay,
immunomodulator.
That's very nice. And I have three things to share for which you are the right person to give your comment.
Yes, sir.
Number one.
Yes, sir.
Drug discovery has evolved over a period of time.
Yeah.
And we know 30% of drugs came from natural origin.
Yeah.
And of course, synthetic we are doing simultaneously.
Yeah.
My first question to you, question in the sense, your comment, particularly you highlighted, when you work for natural versus synthetic, do you think these natural drugs and synthetic drugs, are they complementing each other or competing each other?
Yeah, definitely it is complementing each other, sir.
And
yes, sir.
Yeah. That's what I want. Is it only complementation?
Yeah, complementing each other, sir.
Okay. And repurposing from natural is
Yeah.
easy, or repurposing from synthetic is easy.
Synthetic ACs are always, but
Okay.
the purpose is when we are going for repurposing, we are going to use mainly the computational technology. We can easily predict, but when you want to do some modification, then synthetic drug is easy compared to natural product.
Very nice. And one more thing.
Yes, sir.
Today we hear many ways, artificial intelligence, neural
Yeah,
networks,
Yeah,
all
yeah.
these are coming. They are making their own share of, giving lots of probabilities.
Yes, sir.
Oh, drug discovery, you mean to say even in natural drugs? See, why I am asking this?
Yeah,
India has a repository of number of
yeah,
ICMR,
yeah.
screened compounds.
Yeah,
And
yeah.
so far, nobody has come out with any one D particular. 3 weeks back, we had wonderful session with Dr. D.P. Narayana
Yes, sir.
also. And what do you think? A Indian content kind of, ICMR oriented medicinal plants, we should work out with some sort of this artificial intelligence to bring some meaningful D compounds.
Yes, sir. Definitely, sir. That only during my talk, I told it's not only the natural product research. You have to cooperate with synthetic and medicinal chemists. Then only we can come out with a good drug. Otherwise, it's not feasible nowadays. And the research, whatever they are doing, we are having the molecular diversity is very big molecular diversity in our nation, India. So we are not utilizing properly the research also.
That is what.
They have to do the 100% dedicated work just for publication and patent. That way only the research is moving on nowadays.
Very nice.
That is the main problem what we are doing, just for publication, just for patent. It's not the research.
When Japanese they come out with crystalline compound, we are still left with the medicinal holistic extracts only.
Yeah, that is our main drawbacks.
Yeah. One more thing.
We have to change our strategy.
One more last thing. I was so impressed when you showed a photo, a kind of, this is wonderful. A needle, how to track a needle in a haystack. Ravichandran. Sorry. In Indian system of Sanskrit, there is the avadhanam, sasavadhanam, sahasravadhanam.
Yes,
they need to give a problem. How to bring out a needle in a haystack. I think today we are all stuck in the same thing when it comes to identify a particular compound. It is like a stretching a needle in a haystack. You have given a wonderful example. All the compliments to you and good luck.
Sir. Thank you, sir. Thank you so much, sir.
That's fine, Ravindra.
Yes. Yeah. Thank you very much, sir.
Thank you, sir.
Yes. So any more questions from participants or senior faculties?
I think many of the participants, they have asked the PPT. Okay, I'll send to Dr. Ravindra. You can collect from him. I will send you.
Very good.
Yeah. Yes. So with your permission, shall we conclude the session, sir?
Yeah, no problem, sir.
So Dr. Ravichandran provided in-depth explanation of the basics, scope and state of natural product research, as well as the current status of FDA-approved natural medication development. The presentation established fascinating links between conventional natural chemistry and current techniques for boosting the drug development success rates. Sir covered the various topics, including the drug discovery process, pharmaceutical industry and natural product research, hurdles in the drug development, specifically in the natural product design, flowcharts of the sequence to study the natural products, FDA-approved drugs and the contribution of the natural products in it, and the connection between the traditional natural chemistry and the current modern techniques that will help to accelerate the natural product research, specifically including the molecular modeling tools, structure-based, ligand-based drug designing, and even the gene-based drug discovery techniques. So thank you very much, sir, for all your insightful information that you shared with us today.
Thank you, sir. Thank you, sir. Thank you so much, sir.
Yes. Now I request all the participants to start their videos. So we'll take the group photo. Yes. Sir, can you just unshare your screen?
1 minute, sir.
Yes.
Okay, sir.
Yes. Thank you to our today's speaker and to all the participants of the session.
Thank you, sir. Thank you so much.